This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 110, recorded December 3rd, 2010. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV. Joining me today to talk about viruses is Dickson Despommier. Hey,
Hello.
Dixon.
Hello, Vince.
Right across the desk from me.
Yep.
Nice to see you again.
Nice to be seen.
Excellent. Also with us from the wilds of western Massachusetts, Alan Dove.
Good to be here.
How are you, Alan?
Doing okay.
Been a couple of weeks.
Yeah, I guess it has been. We had Thanksgiving, and good to be back.
All right, good to have you. And finally, last but not least, we always get to Florida last, I guess. Hey, Rich.
Hey, Vincent. Hi, everybody.
Hey, Rich.
Hey, Rich.
Hey,
Good to be here.
Hey, fellas.
Hey, fellas.
I like that.
The gang's all here.
I guess he's not in a fella mood today.
I'm in a fella mood.
What's the temperature down there?
64 degrees.
Ooh, it's chilly.
Actually, the last two nights it was colder here than in Boston or Seattle or Austin, Texas. I keep track of these places because that's where my kids live.
What's that doing to the orange crop?
There aren't any oranges in Florida anymore unless you go way south.
Okay.
There used to be big orange groves all around Orlando, and they finally gave up. It just got too cold.
Interesting.
Maybe now that the planet's warming up, they'll replant them.
Global clothing. Global colding.
Right. So there you go. 64, bright blue skies. Not
Okay.
a cloud. All right.
Well, here it's cloudy. I don't know what the temperature is.
You're right.
All right, let's talk about viruses, shall we? But before we do that, let's talk about bacteria.
Yes, indeed.
Ooh.
Bacteria that love arsenic, or that can grow in the presence of it and not phosphorus.
Not. Wait a minute. How do they make their DNA then?
They
With
put
arsenic.
With arsenic.
With arsenic.
Yeah.
And
So-
so how does that thing behave?
Well, arsenic is chemically very similar to phosphorus.
Well, so is strontium to calcium.
Strontium, if you look at the
Right.
periodic table,
Yeah.
right, arsenic's right above phosphorus. It's kind of the electron makeup is similar.
It's true.
And in fact, that's why arsenic is poisonous, because it goes into all the phosphorus pathways in your cell.
Indeed.
So this bacterium was isolated from Mono Lake.
I've been to Mono Lake.
It's a beautiful place.
It's very interesting.
And they went into the mud, and they got this guy. They actually selected it in arsenic medium. So
Wow.
Right. It's normally a halophile. It normally has phosphorus in its DNA.
yeah, and in fact, it likes phosphorus better. But if you just put arsenic in the medium, it will grow, and it incorporates it into DNA and proteins.
Which is pretty badass.
I'll say, especially if you're a filter
I mean,
feeder.
it doesn't just survive arsenic. It actually eats it. It just
I understand this lake has a lot of arsenic in it, right?
It's
Yeah, I
loaded
think
with all
that's-
kinds
yeah,
of
that's-
heavy metals. Yeah.
So
It's a bad lake.
the authors, of course, are saying this is a new form of life, and others are saying, "Well, wait a minute." I don't think we need to go that far yet. But
I think Craig Venter has set the bar so low for a new form of
that's a great point. But here's my thought. Okay, let's assume for a moment that this is a new phylogenetic tree, not just a branch, but an entire independent evolved life, right?
Mm-hmm.
Do they have viruses?
Ah.
That's my question. Because do you need viruses to have life, or could you have some life without it? See, we only have one experiment of life in viruses here on Earth, but if this were a second one, that would be cool to look at, right?
Absolutely.
But even if they are just, modified bacteria, it would be cool to see if there are viruses that could infect them. They would have arsenic in their DNA, I guess.
I would think that any virus that normally infects this bacterium would probably be able to survive that because it's relying mostly on host machinery anyway.
Sure.
And just think, the virus would be full of arsenic.
That's right.
What could you do with that?
Yeah. I wonder if that
the virus also
could.
Yes,
if
go
that
ahead,
virus
Rich.
infected
Take it, Rich.
if that virus infected normally both arsenic type and regular type bacteria, I wonder if it's got arsenic in its DNA, if it can infect regular type bacteria.
Yeah.
Or goes to pieces.
Then you could imagine it might be toxic, right?
Yeah.
Indeed.
I couldn't get out of this paper. Maybe one of you guys did. It sounds like the DNA is not totally devoid of phosphorus. It's got more arsenic in it than you would expect.
More arsenic than lace.
Yeah, but it's not like completely substituted or something
Right.
like
I think
that.
there's traces of phosphorus, right?
Yeah.
But you can never rule it out totally, I suppose.
And what they did, it's important to note, is this was a laboratory experiment. This is not a naturally occurring arsenic-loaded strain. They grew it in the lab, gave it no choice but to eat the arsenic. And so, yeah, there was a certain amount of phosphorus around from the original DNA of the bacteria. And the arsenic got incorporated to the degree that it could. But sure, I would expect there was still some phosphorus in there.
Yeah, the moment I heard about this, actually, I was asked by the BBC to comment on this yesterday, and I declined because I didn't know anything about it. So, but what I would have said about it,
that
if
stopped
I had
you?
asked, oh, absolutely. It has so many times, Alan, you have no idea, which gives credence to what I've. No, that's not true. I wondered what the utilitarian virtues of this might be with regards to remediation of arsenic contamination.
I'm not sure that it really gets you anywhere.
Yeah, because if it normally uses phosphate, that's true.
And also having the arsenic move into the bacteria. What's toxic about arsenic is any form of it. It's not that you put it in DNA and now it's not toxic. It's still toxic.
Right, but if you could filter out the bacteria as they were being produced in a toxic environment, like say in a lake, where you wanted to get rid of arsenic in some way, this bacteria would sequester it for you. If you could then find a way to get rid of the bacteria, you
just have to find a place that's devoid of phosphorus in order to remediate it.
Yeah, well, the point is that I didn't know that it also used an alternate pathway for phosphorus, but
right. Yeah. Right, its primary pathway, it uses phosphorus.
So do you suppose this guy uses ATA for energy?
Exactly.
Adenosine triarsenate?
Exactly. Yeah.
Wow.
Arsenate, ASO43 negative, and phosphate, PO43 negative, look very similar, and they pump into the same pathways.
Interesting. So that raises another question then. Why haven't all the life forms just taken advantage of arsenic whenever it's present, and instead of phosphorus used arsenic? Why is it toxic? Why is it toxic?
I think there's a lot more phosphorus on Earth than arsenic.
But why is it toxic, though? If you can make DNA from arsenic, why would it be toxic?
They've got a rap about that in the introduction of this, but I can't.
Why is not toxic to the bacteria, but it's toxic to us?
No, to anything. I mean, if you could substitute arsenic for phosphate, like we can strontium for calcium.
The point is that this is a bacterium that can handle arsenic.
Understood.
Right? We can't.
Okay, why can't we?
Well, because it's incorporated into molecules and it kills us.
No, that's not a good enough answer.
The issue is, while they are highly similar, they are not identical, to the point that some of our enzymes are not capable of functioning with arsenic.
Yeah, see, it says here, downstream metabolic processes are generally not compatible with arsenic incorporating molecules because of differences in the reactivities of P and AS compounds.
Right.
But in this particular, how many genes does this bacterium have? It must have as many as a normal bacterium, right?
Yeah.
So its enzymes are not particularly affected this way.
Right. Its enzymes have evolved enough flexibility to be able to deal with arsenic, but there isn't enough selective pressure on other organisms like us.
Yeah.
It's much easier for us to just avoid arsenic.
Right, but this is still a unique finding then.
It is.
It's very cool.
No, nothing against that.
So clearly these guys are in an environment with a lot of arsenic, and they evolved to be able to handle it.
Correct.
Yeah.
Right. And then you can in the lab evolve them further to rely on it entirely.
Yeah.
Yes.
It's very cool.
Well, ultimately, it harkens back to a lecture I once heard from a guy from the University of Indiana, whose name I'm blocking on right now, but who was the first one to actually discover bacteria in the bedrock.
Yeah, right. Tom Brock, right?
No, it was someone else. I know Tom. He actually ended up at Rockefeller, as though you recall.
No.
No? Maybe it was Tom Brock then. It could have been. It was a Harvey lecture, but the point is that he also indicated that all of the ore deposits throughout the world were created by bacterial manipulation of the rock.
Yeah.
Yeah.
I mean, gold, silver, platinum, uranium, you name it. And the reason why it's found in heavy concentrations in these seams was from some microbial action on the rock itself. That's an amazing revelation, if that
so
turns
here
out to
is
be true.
in 2003, a team of scientists has discovered bacteria in a hole drilled 4,000 feet deep in volcanic rock in Hawaii.
Yeah.
This is Martin Fisk, professor at Oregon State University. The story we're talking about just came out in Science Express. It is by Felisa Wolf-Simon and her collaborators. And I have a few links to some interesting stories that are relative to this, so we'll post those.
Good stuff.
It's very interesting. But, of course, this is not This Week in Microbiology.
No.
But that's coming in January.
TWiV?
Stay tuned.
Really?
Yeah, it is. But we thought we would mention this because this is a big deal and everyone should know about it.
Absolutely.
Sure.
But it's not a life form from another planet.
No.
Well, some of the press is kind of playing it. And I bet the BBC wanted to ask you that.
Well, I never got to that stage, believe
It was
me.
actually pretty amusing to me. I saw this under embargo.
Right.
And then, I thought, wow, this is really, really cool. I can't wait for this to come out. I'm not reporting on it. I don't,
Right.
I don't pitch the spot news type stuff anymore. But then somebody broke the embargo and they had to lift it prematurely.
Yeah.
So there is a nice blog post by Ed Yong at Discover Magazine. And he thinks it's great, but he says, this is not another life form.
No.
It's an amazing result, but there is even room for doubt there. And then at the end, he says, this hasn't stopped the hype machine from rolling forward, fueled by a public announcement from NASA, teasing a press conference about an astrobiology discovery. And he said, that's a shame, because this is good science that should go on its own, right?
Yep.
Right.
Then he has an update, which he just added. John Sutherland from the MRC Laboratory of Molecular Biology at Cambridge adds to the skepticism. He notes that arsenic-based compounds are not sufficiently stable in water for the phosphorus to arsenic substitution implied in the paper to be functional. Well, like any observation, it remains to be validated by other groups, right?
Indeed.
Right,
So
sure.
in other words, in a petri dish you can get this to work, but at Mono Lake it might not be true. And nobody else in this group has ever been to Mono Lake. It's really quite amazing to be there.
I was close by this summer, but didn't make it.
No, you're our mono attendee at Mono Lake.
All right.
It has very few life forms, but it has a richness of each of those life forms, including these saline-loving flies that are just absolutely everywhere. And the birds on Mono Lake feed on these things in big mouthfuls. No, there's no fish.
Can't fish?
Vince is indicating that there might be some fly fishing involved.
There's no fish in the lake?
There's no fish in that lake. Oh my gosh, no. It's an
It's a hypersaline
It's an alkaline deposit lake because it doesn't go anywhere. It leads out of the Yosemite National Park into the middle of a desert.
Mm-hmm.
So it just keeps evaporating, and all the salts that wash down out of Yosemite through this little river that leads into it. And Los Angeles used to tap that river for water. So there used to be a big society out in California that says Save Mono Lake. And because Los Angeles was drinking it, and they did.
This explains a few things about Hollywood.
They actually did save Mono Lake from being drained dry because of the thirst of Los Angeles. But it hasn't recovered to its full length yet, or depth, I should say. So you've got these big pillars of inorganic deposits sticking up out of the water, which they formed under the water. And you can see how far down the lake is now compared to what it used to be, simply by looking at how high out of the water these large columns of organic deposits are. It's quite amazing to see them. Yeah.
But we should get to the viruses.
We should get to the viruses.
And our first virus is Frank Fenner.
Yeah, he died.
He died on November 22nd. And I'm holding a book called The Biology of Animal Viruses. It's the student edition. I bought this as a graduate student.
Wow.
And this is a wonderful, wonderful book. My first virology textbook.
I'll be darned.
And I think this was a great book. And I also, so there have been a few stories about Frank. He was an older guy who had done a lot in pox viruses, right, Rich?
That's right.
Do you know him?
I have met him a couple of times, and in fact, some years ago, he submitted a PNAS paper for us. He was one of the few foreign members of the National Academy. So
how would you summarize him?
Brilliant.
Right.
A real contributor in a big way, in a global way. A very nice man.
Right.
And just full of energy. He died at the age of 95, and he was in every day at 6:30 writing up until a year or so or months before he died. Every time his name would come up in conversations, people would say, how's Frank doing? Well, he's still at his desk writing. He was and a pioneer in many ways. He pioneered virus pathogenesis with his experiments with mousepox in mice, and that model, as you pointed out, Vincent, is still used in all the classic texts. I still teach his work from 50, 60 years ago or something on mousepox pathogenesis. He was an important figure in the eradication of smallpox, and was part of that campaign. As a matter of fact, he was respected so much in that they gave him the privilege of making the official announcement of the eradication in 1980. And as part of that program, it's a good thing Frank likes to write, because as part of that program, one of the World Health Organization's mandates was that what was done needs to be recorded. And there's this volume that I have sitting on my shelf that we in the field called The Red Book by Fenner and Henderson, and I'm just reaching up for it.
D.A. Henderson?
D.A. Henderson.
Oh, yeah.
Arita, Jezik, and Ladny. And it is, let me look, 1,460 pages long.
Wow.
Oof. Called Smallpox and Its Eradication.
Right.
And it is a profound reference to the history of smallpox and to what they did in the eradication campaign. If you need to know anything about eradicating a disease, it's here. It's
right.
amazing.
So far we've only eradicated
and Frank
one.
Also, as we've talked about before, was responsible for the experiments that imported myxoma virus into Australia.
Mm-hmm.
Right.
Used that to trash the rabbits. And responsible for following that up and coming to the realization that the rabbits and the virus reestablished an equilibrium situation. So his contributions are profound.
Right.
He'll be missed. He was a great mentor.
Yeah, we have a figure of his in our textbook, which shows the dissemination of mousepox in an animal. Classic studies.
I met him when I was at Rockefeller as a postdoc.
Oh, yeah?
Yep.
He was there for a bit,
He was there briefly.
Yeah. So that you intersected.
We did.
Did he yell at you?
Why would he do that?
No, I'm just saying, because he's apparently a nice guy.
Should have gone into virology, Despommier.
That's right. What are you messing with those parasites?
That's right.
So I wrote a blog post last week I will link to in the show notes. But there you go, Frank Fenner, 1914 to 2010. You going to be 95, Dickson?
You betcha.
All right. You want me to write your obituary?
No.
Why not?
Let's see. Why not?
You want somebody else to write it?
Let me give pause to that. I don't want anybody to write it, actually.
No, that's good. Not just me. It's not
personal.
that's
nothing personal.
Oh, okay. Because we could write it now and you could proof it and all.
If you were a newspaper person, you would actually do that.
Yes. Now. I understand that's how they do it.
That's correct.
Yeah, that's why every now and then there will be one that slips out early.
Right.
Hey, didn't Steve Jobs' obituary slip out like last year?
I don't know. Yeah, I think that happened. Yeah, somebody accidentally hit the press button on it or something.
Mark Twain certainly did. He had a great quote for that one, right?
Yeah.
Yeah. What was that quote? It
says the rumors of my death were greatly exaggerated.
That's right. Very good. All right. The next story is the polio outbreak in the Congo, which is a country in Africa.
Indeed.
And this, I actually received an email. This has been ongoing for several weeks now, and I had received an email from Jay in Australia, who's a scientist at the Polio Reference Laboratory there. So these are global laboratories that routinely sample specimens to see if they have polio in them. So what happens, you look for acute flaccid paralysis. This is the clinical diagnostic indication that someone might have polio. You get samples from them. You send them to a reference lab, and they check them out. And Jason says, looks like the polio outbreak in the Congo is pretty bad. Over 120 deaths, 280 paralytic cases so far. There are actually more, as we'll see. Looks like this variant is related to the type 1 circulating in Angola, which was originally derived from India, most likely from migrant workers. Interestingly, the average age is between 15 and 29 years old, which is outside the usual AFP, acute flaccid paralysis, surveillance guidelines of less than 15 years of age. This is probably due to the vaccination gaps in the past when this age group were less likely to have been exposed to either wild or vaccine virus. Pretty depressing, really. Now, Dickson, why do you think it's depressing?
Because it shouldn't be happening.
And we're so close also, right?
Yeah,
supposedly.
I mean, it reminds me very much of what's going on in Haiti with regards to cholera right now. It really shouldn't be going on.
It's an absolutely inexcusable disease to die from.
100%.
Cholera, you mean?
Yeah.
Cholera.
That's right.
Yeah.
Polio too, right?
Polio too. Yeah, polio should not be occurring. Yeah,
it shouldn't be occurring. But cholera, if you get it, you could treat it, right?
Every
That's
one
the
of them.
point, yeah.
You should be able to.
That's
yeah.
correct.
But the
Clinics
sad reality is that there are places in the world where you can't.
That's right.
So a little info on this Congo outbreak. As of November 22nd, the cumulative number of cases is 409, of which 169, 41% were fatal. That's quite high.
It's very high.
And the age group is...
Is that higher than usual?
It's higher than usual, absolutely. Yeah, it's usually much, much less than that. And part of the reason is probably because this is targeting older people. Usually as you get it older, it's more likely to be lethal. And as Jay said, it's probably because there were gaps in immunization, and so people reached a certain age without being immunized. And
so death from polio, is that from neurovirulence? Is it like paralyze your respiratory system or something like that? Or is there death for other reasons?
It can be paralyzing your respiratory system, but could also destroy other nerve centers in your brain, depending on where the virus is replicated.
Remember the iron lungs that people used to put into?
Yes.
So
that's a certain
exactly
fraction.
Right.
That's the bulk.
But it's the neurovirulence in one fashion or another that kills you.
Yeah. Yeah, either making you not able to breathe, it stops your heart, or other neurological reasons causing death. Now, interestingly, so it says here in this ProMed Mail report, this is one of the largest wild-type 1 polio outbreaks ever described, associated with an unusually high case fatality rate. So they've got... Yes, Dickson.
Well, I'm just curious as to whether there might be some other confounding factors here, because this is occurring now in a less developed country, and perhaps then it's been ravaged by civil unrest for many, many years. Maybe there's some nutritional aspects to this, or environmental aspects to this, or maybe another infection superimposed on top of this that might be an underlying cause for the high mortality rate. That's all.
It's possible.
Thinking out loud here.
We don't have any evidence, and I don't know, from this health system, I don't know if we will get it. But it's
going to be very, very hard to get data from a place like that.
Yeah, exactly right.
Now the last,
just to put things in perspective, the Congolese civil war that's going on there is one of the deadliest wars in history.
That's right.
I just looked up the latest stats on it, and apparently over 5 million people have died in this war that's been going on for more than a decade. So the place is really a mess.
Total chaos.
So you're right. There could be other confounding issues as well.
Sure, I mean, you come in with a stressed person to begin with, they catch this disease, their immune system is way, way down, and the next thing you know, they're dead.
Polio was almost eradicated in the Congo. Last case in 2000. 10 years without polio. And then, boom.
Boom.
Yep.
You introduce it in. Now, they have some lab studies related here. So they had tried isolating viruses from a number of people. They finally turned to PCR. And then they they had 19 samples. 17 of the 19 were positive for wild-type 1 polio virus. Basically identical to an Angolan strain, as Jay had told us, which originally came from India. Now, interestingly, there was one other virus present in one individual. And this was a person who died, actually, one of the fatal cases. They found enterovirus 109. So beyond polio and Coxsackie and echoviruses, they start to number the enteroviruses, 70, 71, 72, and they've reached past 100. Wow. We don't know much about these. We just have sequences. So this gentleman had entero 109, one of the highest enterovirus RNA copy numbers ever described, and they didn't find polio in this individual.
What did he die of?
Exactly.
We don't know. Doesn't say here. A deceased patient. So It could be that this patient had paralysis and was identified as part of the outbreak. It was sampled. This is a rectal swab, and then he died. They don't actually provide the clinical information. So we don't know if this virus killed him or if something else did.
But they found this because he was paralyzed, so he came to medical attention, and they assumed it was polio, and then they found EV109.
Okay.
So whether EV109 paralyzed him, who knows? It's just there. So
if this is a virus that came through Angola from India, I wonder if there's enough data on this in those other populations to address Dickson's question. So if there were outbreaks due to this virus in other populations, I wonder if they had the same sort of mortality rate.
Right. What was the case fatality rate in India?
Oh, it's not this high at all.
I bet you it was lower.
Not at all. I'm trying to see if there's
Surprisingly, their healthcare system isn't so bad. There are a lot of regional hospitals and laboratories and healthcare workers working all over the place. I mean, it's certainly not like ours, but it's commendable to see what they've done with 1/4 of the world's population.
Yeah.
There was one other point I wanted to make. Yes. So what do we do? Dickson, what do you do when you have an outbreak like this?
Well, Jesus, I would run right in there with a bunch of vaccine and start immunizing like crazy.
That's what they've been doing, immunizing several million people.
Absolutely.
And this is, WHO
trying to put the fire out.
So at least 1.1 million people are to be vaccinated. The epicenter was just Point Noir, and a further 600,000 people in neighboring regions. So we'll see. That should quell it. These things don't go on forever.
Nope.
Because you get. Hey, by the way, this is what we forgot. If you have 400 cases of flaccid paralysis, how many actual infections are there?
Gentlemen,
4,000.
Nope.
Is that right?
40,000.
40,000.
Right.
Ooh.
I know.
So a lot of people walking around with this virus and you don't know it.
Yeah.
That's why it can spread readily.
Right.
But that immunizes the population eventually enough to stop the spread of the virus. Mm-hmm. So this is actually good that it doesn't cause a lot of disease, right? Self-limiting.
Right. The virus is to some extent its own vaccine. It's just a really bad vaccine.
Sure. Remember, though, the take that back in the 50s when it was epidemic throughout the United States and the Northern Hemisphere. Everyone thought it was a very rare disease because it causes paralysis, and we didn't actually know how many other people were infected.
Yeah.
Turns out that almost all of us were infected.
Yeah. That's an interesting point. The amount of paralysis was not huge in terms of disease burden in the U.S., and the case has been made that this was a project of the national foundations fueled by FDR's paralysis, and that there were really other health needs that were more pressing, but that's a historical opinion.
It's a high-profile disease, though. I mean, the disease itself is really devastating.
Right. It's pretty hard to ignore paralyzed kids.
Right. And then you had cases of partial paralysis also.
Right.
Sure. Yeah.
So
some people still have
that's correct.
Lingering limb weakness and so forth.
Exactly.
Okay, the next story is molecular. Oh, this is, I think, from Alan, right? Molecular evolution of HIV solves 2 criminal cases.
Yeah,
this is very cool.
Came across this in my usual stack of press releases and such. So this is a lab. Let's see, where are these folks? They're at UT Austin and Baylor, and they did a phylogenetic analysis for 2 criminal cases, and these are these cases. Are 2 individuals, Anthony Eugene Whitfield and Philippe Padu. And these are guys who allegedly, well, I guess now that they're convicted, we don't necessarily have to say allegedly, but they deliberately spread HIV to as many women as they could. And they were charged, totally separate cases. They're in different states, but I think they were both charged with assault with a deadly weapon.
Right.
HIV being the deadly weapon. There is legal precedent here where people have used phylogenetic analysis to either rule in or rule out particular theories about where the virus came from in a particular individual. Probably the most famous is there was a dentist in Florida who didn't use correct precautions and transmitted HIV to a bunch of patients. And so they they were able to determine that there was a pretty close relationship, or a very close relationship, between the HIV sequences in half dozen of his patients to the HIV sequence in him.
So was he convicted for that?
He was convicted for that.
But different charges, though. This was not intentionally, right?
Yeah, I think that one was not intentional. And then there have been other cases, again, accidental transmissions, which presumably end up in the civil courts. And there have been accusations where patients said, "Oh, my doctor gave it to me," and then you can do the phylogenetic analysis, and you could say, "Well, no, that's not really possible because your HIV-1 is not closely related at all to the one that your doctor has." So this has been used before, but one of the problems, and one of the things that I presume defense attorneys would bring up in a case like these, where there are felony charges and serious time on the line, is that you can't, with these plain old phylogenetic analyses, all you can say is how closely related one virus is from one individual to another individual's virus. And HIV-1, as we've talked about on the show, mutates a lot, like a lot of viruses. So the virus in one person's bloodstream is really going to have a whole bunch of different sequences, and they're going to converge on a certain general area, but it's going to be a certain amount of spread in the sequences that are there. And then you look at somebody else and you say, okay, well, they have this whole set of sequences, and you look at where the overlap is. It's not a definitive, this person gave it to that person type of answer. So what in this case, the other thing that was different is I think in most of the previous cases, the analyses were not blinded.
Right.
Right.
So the lab doing the analysis got, okay, here's a sample from the dentist. Here are the samples from the 6 patients. Tell us how closely related they are.
Right.
That's bad.
That's bad. That's not, that's not what you want in science, and it's certainly not what you want in the courtroom. So in felony cases like these, they had to take things a step further. So in this case, they did a totally blinded analysis. The district attorneys and the researchers did not know which sample was which. They just had them all in coded tubes. And they did complete phylogenetic analyses of all of them. And they also worked on the assumption that when HIV is transmitted, and this is fairly well backed in literature, when HIV is transmitted from one individual to another, it goes through a genetic bottleneck, because only one or two viruses, individual viruses, will infect the next person. Mm-hmm. So person A has an infection of HIV, and they have a Sagan-esque billions and billions of HIV-1 particles circulating, or genomes in their system. But then only one or two of those are going to get to the person who they infect, which means that those founders, or that founder, will then be the parent of the entire population of HIV in that recipient. So then, using that, you can say, okay, it's not going to be just any old overlap. We're going to have an actual subset. The virus in the recipient should represent a subset of the virus that was in the source.
Right. So without that bottleneck, you can't really determine this transmission, the directionality.
Exactly.
Right.
Right, because you could say, okay, there's overlap between the two. All we know is maybe they got it from a common source. Maybe A gave it to B. Maybe B gave it to A.
Mm-hmm.
Right.
And then you'd have to go into other evidence to figure out which, you might be able to figure out date of infection, but that could be a little dicey. So I noticed, Vince, you made some notes about some helpful definitions here. Maybe you'd like to walk through those.
So, yeah, in the abstract to this paper, they say, during transmission of HIV, a genetic bottleneck occurs resulting in the paraphily of source viruses with respect to those of the recipient. Now, they don't tell you.
I had to look this up.
You have to look it up. Even I did, because I don't use paraphily. on a daily basis, so I thought it would be useful.
I don't even use it on a monthly basis.
Yeah, well, now you got it this month.
I think the last time I used it was in a genetics class, and many years.
So here are the definitions. Monophyletic means common descent from a single ancestor.
Correct.
You and I are monophyletic, right?
Indeed.
If there is a group that is made of a common ancestor, like humans and all its descendants, that group is monophyletic, which is from the Greek of one race. Now, a group,
and going back to the HIV example, the HIV in one patient should be monophyletic from the origin virus, from the founder virus.
Now, if a group contains a common ancestor but leaves out some of its descendants, it is paraphyletic, and that's what the phase was used here. All right. So if the guy A gives it to person B is going to have only a subset of what was in A, because of what Alan said, that you have this bottleneck. So that's what paraphyletic means. You don't get all of the descendants from the person who infected you. And by the way, all living things on this planet are thought on the basis of DNA analysis to be monophyletic.
I was going to say, how would you ever use the polyphyletic in a
Viruses are polyphyletic.
Okay.
Well, and it's a question of context, where everything with DNA is monophyletic if you go back to the first organism.
Right.
But you could say that all badgers are monophyletic from a single ancestor badger. So you could say within that context, and all HIV-1 particles in individual A are monophyletic from a common ancestor. But
so let me ask for a point of clarification here, as the only non-virologist in the crowd. When it goes through the bottleneck, it's a single
one or two
make
it.
Yeah, well, let's say 2 make it.
Yeah, you can get infected with 2. Yeah, one or 2 though. You don't get infected with 10 or 20.
No, just 2.
1 or 2. I mean, you probably get a bunch coming in, but most of them fall on fallow ground.
Okay, so they're diphyletic.
Well, here's an example in the definition where I got this from. They say warm-blooded animals are polyphyletic, but that doesn't make any sense biologically.
None.
It's useful to talk about warm-blooded animals in some contexts, right? And they're polyphyletic, but biologically it doesn't have any meaning.
None.
Right? Okay.
Just means they're different species that are warm-blooded, that's all.
Yes, exactly. Yes, Rich.
I pasted in a wiki link there into the notes that has a really nice diagram of the difference between monophyly, paraphyly, and polyphyly. That's helpful in understanding what this is. I found that useful.
Yep.
So the bottleneck, among other things, has to do with the tropism of the virus, correct?
Mm-hmm.
Right.
That the infection usually happens in macrophages because they're the handiest susceptible cells around, and the virus that infects macrophages it's called M-tropic because it's tropic for macrophages, and the co-receptor there is different than ultimately when the virus establishes itself, it starts infecting predominantly T cells instead of macrophages, and that involves evolution in the infected individual of a T-tropic virus that's using a different co-receptor.
That's right.
And so the bottleneck is not only just in terms of numbers, but it's genetically, typically a specific subset of the virus that's in one individual. At the same time, not all of the victims, if I understand this correctly, are going to be infected by the same virus initially.
Right.
It may all be predominantly an M-tropic virus, but genetically, say, a variant from the M-tropic virus existing in the original donor, if you like. But that bottleneck, nevertheless, is important, and I guess restricting the diversity of viruses in a fashion that allows you to determine the direction.
Well, the donor will have all of the sequences of the recipients.
Yes.
They will have different sequences, but they will all have come from 1 or 2 viruses from the donor.
Right, but the recipients will not have all of the sequences of the donor.
Right.
Yes.
And this is also why HIV transmits very, very poorly, really. I mean, it's a global pandemic, but the chance of getting it on any particular sex act is relatively low. It's the cumulative, you keep keep pulling the trigger, and eventually you get to a chamber with a bullet in it.
Right.
So yeah, I mean, these were cases. These two guys, one of them deliberately tried to transmit HIV to 17 different female partners through unprotected sex, and they say he engaged in more than 1,000 sex acts with these 17 women over a period of time, and 5 of them tested positive for HIV during that time.
So they basically got samples from all these people, including the defendants. They blinded them, and they were all handled very carefully according to chain of evidence. Baylor, who did the sequencing, got one sample at a time, did the work, and then got rid of it to prevent contamination.
Yeah, the description of the precautions that they used is phenomenal. It's amazing.
Then they said, they took the data, they did the analysis, and said, here is what we think is the donor. And they gave it to the court, and during the trial it was revealed. They opened the envelope, and the guy that was sitting at the defense stand was the one that the labs had picked in both cases.
Perry Mason couldn't have done it better.
I'll tell you, talk about this would actually, I can see a TV show or a movie about this. Talk about court drama. It's amazing. CSI, right?
Right.
So this was quite dramatic, I would think, and they got it. And now, one cool thing that I got from this is that the farther you get from the infection, but they say the paraphyly, with respect to source sequences, is expected to decline over time. So the farther you get away from the infection, the less convincing the data are going to be. And in fact, 2 of the people in this case...
How do you prove premeditation here, though?
Well, that's for the court. That's not
that's
science.
That's actually a very good question.
They say
Dickson,
something about it, right?
They say something about that. They say they're very careful in their paper to say that this implies nothing about motive.
Right.
All we can do is tell you where the virus came from, where it went.
That's right.
It's up to the court system to deal with issues of motive.
Right. The virus doesn't care about motive, and we're studying the virus, so we can't tell you anything about motive.
That was very interesting. So I think lawyers read this draft, don't you think?
I would expect. I would guarantee you that at least 2 lawyers read it.
I know how to make a fortune now, guys. Just go back to law school. You can be the interpreter for all these new coming data.
This paper is actually what's called an inaugural article. So when someone first gets into the National Academy of Sciences, they write an article like this. So this is actually a summary of this person's work, and these papers describing these results have actually already been published. So this is sort of a review of it. But I assume it was all reviewed, because they even say in the bottom, these trials are finished. There's no more financial things going on here. We're all okay. We can talk about this.
There's no obligation.
It's clear that this was written with some sort of legal oversight into the whole. And the supplementary pages are as interesting as anything else, where they describe the actual court case and stuff like that.
Yeah, it's a very interesting story, and it's very educational.
I could imagine, though. I'm really thinking out loud here, but I think you guys see the same thing. This done well in a movie, you could educate an entire public as to what DNA is, what a virus is,
it won't be done that well.
How HIV is transmitted, how it impacts society, and how you deal with this.
I agree, but it wouldn't be done that well. For example, Dickson, in the discussion, very interesting discussion here. The recent enactment of national laws that criminalize transmission of HIV in more than a dozen African countries has led to considerable controversy. Here, and I want you guys to tell me what you think. Some argue that criminalization of HIV is likely to have a negative impact on public health and human rights.
Well, only because people who have it are less likely to come forward to get treated, because there's some stigmata associated with that then.
And so that's the public health impact. What about the human rights impact?
Well, I suppose if somebody is somehow prosecuted or denied some privilege because of their HIV status, and in particular because it's been criminalized, that could infringe on their human rights.
Right.
Okay, I mean, not everybody... It's not your fault necessarily. You got HIV, and all of a sudden, you're potentially a criminal for passing it to somebody.
Yeah, that's exactly right.
So many states have passed HIV laws like this. But of course, there are other viruses you could transmit that are lethal, right? I guess because not the biggest numbers are with HIV, we have laws for that, right? It's kind of the easiest. It's sexually transmitted, so easy to transmit without, you don't need a needle. You don't need it's not easy, but you don't need any equipment except yourself. And you don't need a needle. You don't need bottles of anything. You don't need contaminated food. So I suppose that's why.
It's a combination of HIV exceptionalism and also the biology of the virus. It's a long-term chronic fatal infection that is spread sexually. Right. So people have a lot more time to find out that they're HIV positive and to decide whether they're going to, tell anybody about that and so on.
Right.
But there's another virus sexually transmitted which can kill you, and that would be HPV.
Sure.
And so why don't we have laws criminalizing that? Maybe it's just a matter of time.
Yeah, as I say, part of it is AIDS exceptionalism. It's this virus is treated differently from similar diseases.
One other thing I liked here is in the beginning they talk about why DNA forensics works for humans, because our DNAs are so similar, right? But for viruses like HIV, it's a different story. So you depend on sequence analysis, phylogenetic, and bioinformatics.
Right.
Right. It's not just a simple yes or no.
Right.
But so this hasn't made it into CSI, I guess, the phylogenetic trees. But I think it would
I'll
make
bet
a
you
good...
it will. I'll bet you it will in the end. It'll be interesting to see.
Well,
By the way, this being a PNAS article, our listeners, that's free online, right?
Yep.
Yeah,
so
this one.
our listeners can read
This one I actually had to sign in to get to.
Yeah,
is that right?
Yeah, this is.
They're not all free online. PNAS is a mixed publication.
Which I must say, that's bad. This is our government agency,
Your tax
right?
dollars at work.
This should be free.
No kidding.
Write your congressmen and senators.
Especially something like this.
No question.
This has such broad public implications. It's terrific.
This is
The
true.
National
and
Academy-
interest.
Why isn't
It
this
may
free?
wait a second here. This is early edition. This is online before press, which I would think they would release publicly faster, but I don't know if they have a separate protocol for that.
But even though there are some articles you can't access, and still, I think that's fundamentally wrong for a government agency, right?
Yeah, the National Academy is a right. They're government constituted. They're kind of in a gray area, but yeah.
Okay. Right.
It's not entirely. I mean, it's not like MMWR,
Right.
Morbidity Mortality Weekly Reports, which is an entirely government-funded publication.
So, what part of this is not government?
The economics,
authors.
the authors, but also I know PNAS for a long time. They have operated on a page charge basis.
Mm-hmm.
I'm not sure who actually handles the back end on the publishing and whether they've outsourced any of that. It may not be entirely simple.
Okay.
But yes, I agree with you that something that is in this journal should be public, and particularly a paper with this much public impact.
Okay. Anyway, that's a very cool paper. I had
Yep.
not seen it. That's great. And it's a really neat story. I agree it could make a good movie, but I don't think it would be done right.
Well, we'll be the advisors. We'll put TWiV out for a bid and see which movie maker wants to hire us as their consultants.
I think the researchers on the paper should probably...
that's true.
a good idea.
They never ask those people. Come on. You could even have a life story of some famous person being made into a film, and they always get an actor for this. They never get the actual person.
Yeah, the actors are better, I guess.
Yeah, right. I can do you better than you.
It's all about fame and famous people, you know.
I don't know. I'd probably want me played by somebody else.
Sure. You don't want to play yourself. Who's going to
No.
But Dickson, who would play you?
Nobody.
What actor?
Nobody.
Come on, there has to be someone.
I mean, if I had to pick somebody to play
pick
me,
someone
I'd
to
pick
play.
Sean Connery.
You
I was-
would, you
Sure.
would, eh?
Absolutely.
Can he talk like you? Can you talk like him?
We both have similar swearing patterns when we get angry.
Ah. Our last story is a commentary in the Journal of Virology called Order to the Viral Universe. I learned about this paper because it's one of the few where the comments were opened up in the journal. Apparently, it generated so much controversy that the editor-in-chief, Lynn Enquist, decided to allow comments. So you could go there. It's like a blog post. You could go and comment, and there's been a lot of activity. So I don't know if the comments are accessible.
I didn't see the comments on what I didn't know that was the case,
Yeah,
so I didn't see those.
I'll check it out, see if it's...
I can imagine it generated a lot of comments.
So this is by Dennis Bamford and Mart Krupovich. Basically, the idea is there are a lot of viral sequences, and now we're classifying viruses mostly by comparing sequences, which is something we discussed. Remember Elio Schechter's comment last time about doing that? And Dennis says, sequence comparisons are not the best for putting order to the viral universe. We've got more and more viruses. We're losing track because the sequences don't go far back enough, and the viruses are all mosaics. You get all this horizontal gene transfer, so you can't really make sense of evolution, actually. So how do we deal with this? Well, you could make network diagrams to display all this, but that's pretty hard. Or you could pick another gene, pick one gene that's unique to a virus, and use its gene product, not the sequence itself, but the product itself, the protein. So he says the best thing is a capsid, because capsids are unique to viruses. And he has this very nice picture of a DNA virus, PCV1, and a plasmid, which have 1 and 2 genes, but the plasmid, of course, doesn't have a capsid. And so this is what makes a virus is the capsid. So he says we should use the capsid structure to classify viruses. And so he uses as an example of doing this the large double-stranded DNA viruses that have a structure called a double beta barrel in their capsid proteins to build their virions. And he says when you look at this motif, this double beta barrel structure, you can classify the viruses, and you can put viruses of prokaryotes, eukaryotes, insects, anything. You can classify them all together, not separated artificially, as you would by sequence. And so he says we should use the capsid structure. So that's what this is all about, using the capsids to classify viruses.
So it's almost like doing a morphology study.
Yeah, in a way.
In a way, sure.
Yeah.
So, Rich, you wrote the classification chapter for fields, right?
and I have to do this again. When's our deadline?
March.
Oh, dear. Oh, brother.
So what's your view of this?
As a matter of fact, yesterday, inspired partly by this and partly by the increasing anxiety about revising this thing, I wrote to the current president of the ICTV, the International Committee on Taxonomy of Viruses, to make sure that I was going to be up to date for this chapter and to ask him a couple of questions. First of all, let's start with sort of the good news of this. There's a couple of things that these guys are trying to do that I'm sympathetic with. Some of their arguments make sense. I'm sympathetic with the idea that you can have viruses that should be grouped together that have vastly different ecological niches, but probably have some common ancestry. One of the things they're arguing for is to have higher taxons than currently exist. Right now, when the
well, nobody's in favor of raising taxons.
Oh, good. Yeah. Good.
Alan,
when they started filling this taxonomy, they didn't start from the top and move down. They started from genera, and then they found that you could define species within a genus, and then they grouped genera of apparently related genera into families, and then to a limited extent, but not exclusively, they've grouped some families into orders. Okay, and they haven't gone any higher than that. One of the things that these guys are arguing is that there may be a way, perhaps using capsid structure, to create higher taxons, and in fact ultimately approach this problem of whether all viruses come from some common ancestor or whether there are multiple independent evolutions of viruses. And my guess is
are viruses monophyletic or polyphyletic?
Exactly.
Yep.
I was thinking about this as we were having that discussion, that this issue was going to come up again. So I'm sympathetic with that goal, but even they point out weaknesses in the problem. First of all, I'm not sure it's really true that we're doing everything by sequence now. And this is one of the things I need to check on for rewriting this chapter. One of the unique things about virus taxonomy is that very early on, when people tried to figure out how to do it, they couldn't decide which characteristics they should focus on or which were more important than any others. And so they decided, well, we aren't going to make that decision, and they used what they call a polythetic approach. So you don't have to have one thing in common to be part of a taxon. Multiple characteristics are considered when viruses are grouped together. And then they said to themselves, well, can we create some sort of algorithm with a whole bunch of different characteristics and computerize this whole thing? And they decided, no, that's too complicated. So what we'll do is we'll just have committees.
Right? That's less complicated.
So much simpler.
Stop it.
But the fact is it works. So you have a committee that does pox viruses and a committee that does these. And then all these committees, when somebody says, well, we got a new genus here and we think it ought to be part of this family, then the various committees that are nominated to be to consider that genus will consider that. And the fact is, it works reasonably well. It's described by one of the historical secretaries of the society, a guy named Regan Mortel, I think, who ultimately came up with the species definition. He calls the process, I love this, the opinionated usage of data. Okay?
That's right.
So I'm not sure that it's all genetic now. I mean, you got capsid morphology, replication style, whether or not it's got an envelope, ecological niche, all this kind of stuff. Certainly the genetics has become more and more an important component, but I think it still is to a large extent polythetic. The other thing is, the capsid thing may work for some groups of viruses, but it's not going to, I don't think it's going to be global.
No.
Case in point, I think this is one of the most interesting examples for virus taxonomy. What do you do with RNA retroviruses and hepatitis B virus? Are they right now they're in different families. If you were to create higher taxa, would you put the RNA retroviruses and hepatitis V virus in the same taxon? Because after all, they're both reverse transcribing viruses.
Yeah.
Which is, you would think that somewhere there's a common ancestor for that. Well,
it depends. If the RT came from the cell, then it's irrelevant. And that's one of the arguments he makes, that most of the genes other than capsid encoding genes probably originated from cells, so it doesn't make any sense to use them to classify the viruses.
Okay.
I don't know. I'm not saying I buy it. I'm just relating what he has said.
All right.
But he also says that the enveloped viruses or the pleomorphic ones are not going to fit into this either. So this idea, there's going to be a lot of viruses left out of this idea, the pleomorphic. Yeah. So I suppose that when you got down to it, you could look at the nucleocapsid protein in those viruses and ask whether they can be sorted out based on the nucleocapsid protein.
Doesn't this then answer part of the question as to what is the difference between viruses and all the other DNA or RNA containing life forms on the planet? I mean, you're trying to fit big, giant square pegs into very tiny round holes when you try to silo each one of these groups of viruses into categories, because you'll find exceptions in every single group, right? Whereas if you go back to the life forms that we would all agree are life rather than not life necessarily, but something not taking advantage of another life form, by being totally dependent on the machinery of that cell in order to replicate, it's very, very likely, just from an outsider looking in, from a parasitology standpoint, that these things probably arose multiple times in multiple places in multiple organisms, and then don't have these commonalities. But rather, because of the constraints of the ecological niche, they assume the best forms and best shapes, because form follows function. And if form follows function, then it's obvious that these capsids have similar shapes, even though they're not related to each other by phylogeny, because they have to fit into a certain ecological constraint. Like all the phages that infect E. coli have these beautiful diamond-shaped things with little legs on them, and etc. It's just a random. It's not a random thought. Listening to your constraints of trying to put these things into a category, some of them may not have categories.
Well, I think that's one of the issues here, is that classical classification separates into the three domains of life: viruses of eukaryotes, bacteria, and archaea, or prokaryotes and archaea, if you wish. And that's not right, because there are a lot of relationships. He's shown these in these big DNA viruses. He also says the tailed double-stranded DNA viruses of bacteria, archaea, and eukaryotic herpes viruses all have similar structures, so they shouldn't be classified separately.
Right.
They're viruses, right?
Yeah.
So I view this as not a final word, but he's just trying to inject some conversation.
Some logic.
Right. He says, let's talk about this, because the way we're doing it doesn't make sense. And let me just tell you one thing that has happened in classification that I happen to know about, because it's in the picornaviruses. We used to have the picornaviruses. We had enteroviruses, viruses that infected via the enteric tract, polio and Coxsackie viruses. And then we had a separate genus, rhinovirus, viruses of the picorna family that you inhale and infect your lungs. These are now put together in the enterovirus genus based on sequence similarity. Right. The idea was rhinos are similar to polios on the sequence space, so we're going to classify them together. It ignores the pathogenesis.
You lose the richness of the difference.
So does that make sense? And the people, I was at a meeting a few years ago, and the people who wanted to do this says, just live with it. Just deal with it. Yeah. You're old-fashioned. You don't know what you're talking about. But it's not the case. I think you need, as Elio said, you have to consider what's happening now, not just the sequence, right?
Yeah. Well, and this is a duality that's affected microbiology since before Pasteur. I mean, you had smallpox and great pox, which were classified as diseases based on the size of the pockmarks that they left. Sure. Well, one is smallpox virus and the other is syphilis.
Right.
It's completely unrelated organisms.
Yeah.
But it was, this was the way diagnosis was done, and so we think that it's superior to go to a system where you're classifying these things as bacteria versus viruses. I would certainly agree with that. But then how far do you carry that? Do you say that the picornaviruses and the rhinoviruses shouldn't be separated by their diagnosis? They should be brought together because of their genetic sequence similarity.
I would almost be willing to conclude that these are not life forms due to the fact that you can't classify them by some rigorous phylogenetic thing that every other thing on Earth has fallen into, and everything agrees with those. Yeah. And here's the one outlier group that you just can't do anything with.
I would beg to differ on the bacteria. There's a lot of debate now among bacteriologists about what exactly is a species of bacteria. Mm-hmm. Because they do a brisk trade in genetic material. And, at what point does this particular bacterium on your plate turn into a different species simply because it's picked up enough plasmids and transposons and phages? So they don't fit cleanly into a classification system, and I suspect the reality is going to be that nothing that we consider life is going to fit really cleanly into a classification system.
Well, why should it? This is
artificial.
This is just artificial. Exactly. It's for our purposes, for studying.
It's like your computer doesn't actually have files and folders.
Yeah.
But that's not what's going on behind the scenes. That's
I understand that, but somehow you have to be able to teach this to the next group of people.
So,
and
of course.
How do you teach this with there's
a classification system? But if you come up with an artificial classification system, and then you say viruses are not life because they don't fit into our artificial classification system, I think that's kind of tautological.
So one of the things I like here is if you look at and I think we ought to put this link into the show notes, the official webpage of the International Committee on the Taxonomy of Viruses. Having read some of the stuff that's been published by this group and others on taxonomy, I can appreciate that they have a deep appreciation for exactly the content of this conversation, how complicated this is.
Yeah,
so they're open.
Yeah, you get 3 taxonomists in a room and you get 5 opinions.
Right. So their
subsets.
opening statement here is, taxonomy lies at the uneasy interface between biology and logic.
Right.
It's beautiful.
Right, that's right.
The processing of information follows somewhat different rules in these two systems, and the role of taxonomy is to reconcile them as tidily as possible.
Beautiful.
Yeah,
it's wonderful.
Right. That's right.
Just wonderful.
I like the, that's great. I like the opening sentence in the Bamford paper. It is perhaps human nature to seek order in everything that surrounds us.
You know, of course it is, but look at what's going on in physics, for instance, and how much money we've spent trying to bring order to chaos with the Super Hadron Collider and all of the other accelerators that we've spent billions and billions of dollars and millions of man-hour energies trying to figure out what the heck is the basic units that all this stuff is made out of, right? That's an attempt at unification at the highest level, from the universe to the particle physics. It has to all make sense someplace, we think. That's what our matter tells us to drive towards in our philosophy of life, so to speak. And I just had a conversation. I know this is not off the subject, but it's going to take up some time on this, so you might want to cut it out afterwards. But one of the students I was interviewing today for medical school and for the MD PhD program, his mother was a professional mathematician. So I began to tell this kid that I had actually sat at a dinner once, sponsored by the publisher Springer Verlag, at which the keynote speaker was this guy Mandelbrot. And he got up and showed these wonderful iterations. The kid was sitting there and saying, who was Mandelbrot? And I said, oh, wait, I can show you. So I went to the screensaver, and everybody's familiar with it. And Mandelbrot regaled us with this. This was the 25th year anniversary for Springer Verlag in New York. And on the screen, you see this gigantic black figure with all these little parts to it. It's very symmetrical. And then he starts to click, and every time he does this, it just expands that world until you're finally wrapped up in all these spiral figures. And then I turned to him and I said, well, where the hell do you think Mandelbrot got this idea from? Where do you think the idea for the Mandelbrot set comes from? Or where do you think E equals mc squared comes from? Or where do you think Archimedes, looking at the circle and the radius and the diameter of the circle, and says, if I divide one thing into the other, I might get a number. Where do you think that comes from? Okay, so I turned to him and I said, of course he didn't have an answer for me, and I wasn't expecting one either, but I said, we are composed of all these things. And so inside of us, all this matter is running around generating these words and these thoughts. And it's illogical to think that we could come up with anything else but the field of mathematics that then turned out to express the very world that we live in. Right. So you look at something like trying to pigeonhole viruses into groups and things like this, and it's just a propensity of life itself to want to understand itself. And I understand the urge to do this. And this is as confounding to me as an unvirologist as it is to a physicist, wondering how I can reconcile particle physics with planetary and astrophysics. It's it may be at that level that you're trying to find order.
Well, it's all about trying to understand how things work, and to do that, we try and make order and look for patterns.
Order
right?
is out of chaos.
So that's what this is
about. But
what?
Chaos has order. That's the problem. That is chaos theory. It turns out to have high degrees of order associated with it. So the word chaos is not really what we think it means.
So I have my own little. I'm going to quote myself here from the chapter on this topic.
Okay.
I say that. And you can bash me if you want, but ultimately all of nature is a continuum, and the business of taxonomy has the obligation of drawing boundaries within this continuum, an artificial and illogical task, but necessary nevertheless.
Good. No, I love that. I love that.
So actually, I laid that one on a taxonomist at one point, and he griped about the notion that all of nature is a continuum.
Yeah, right.
I guess if you're a taxonomist, you're used to
griped about.
It turns out he was a creationist, Rich.
Well, people can be divided into two types: those who classify people into two types, and those who don't.
Well, there are three kinds of people in the world: those who understand math and those who don't.
Yes.
Good, Vincent.
All right. Let's move on to email. That's good.
Oh, we're going to get a lot from this one, I guarantee you.
Taxonomy is very contentious. I can imagine those meetings.
Oh, they must be horrible.
All right. The first email is from Jaakko, Jaakko, who writes, Dear TWiV hosts, a few weeks ago I made a search in iTunes for XMRV and found your podcast. That's interesting.
Cool.
I listened to one episode on that topic and I really liked it, so I decided to listen some more, and before I knew it, I was hooked. I really like how you can keep the conversation so natural and casual, still being very educational and interesting. Well, we're just infectious, aren't we?
I tell you.
Now I finally reached the last episode and have to get used to just one TWiV a week. Here are 2 articles about a possible connection between swine flu vaccine and narcolepsia in Finnish and Swedish children. They are Google translated, so it's a bit rough read, especially the Finnish one. Basically what they say is that there's been a rise in narcolepsy in children 12 to 16 years old during the last spring. The vaccine linked to this possible connection is called Pandemrix. From what I can tell, the connection is made only via the close timing between the vaccination and the children getting the disease. Also, in the articles, the doctors emphasize that they don't still know if there is any connection between the two. Nevertheless, Finnish health officials are now considering if they should stop giving Pandemrix. So since these articles came out, and this is quite old, it's been shown that there is no link between the two. It's been better studied, so that just was an association. I would also like to suggest a tip of the week. It's called the Khan Academy and is basically an online school for everyone. Totally free, consists of over 1,600 videos covering math, physics, chemistry, some biology and economics, and even a little bit of history. The guy making these videos is called Salman Khan, and his education style is very accessible. And that's not all. The site also has a browser-based math application which covers math from first grade to most of high school. You can find it at khanacademy.org. Lastly, I just wanted to thank you guys for doing this every week. Having CFS, listening to stuff, is sometimes all I can do, so finding your podcast and after that some other very interesting podcasts has brought so much interesting content into my life.
That's nice.
Jaakko from Finland. And he has a virus joke for us.
Uh-oh.
A virus goes into a bar and orders a drink. The bartender takes a good glance at him and asks, "Are you sure you're old enough?" To which the virus replies, "Are you joking? I'm so old that you could even call me retro." Laugh, cry, hear.
Ba-dum-bump.
I appreciate the sending in jokes. I know jokes are difficult to make up. All right, the next one is rather long, so I will summarize it, but we'll post the whole email online. But I just thought this would be of interest to some listeners. This is from Tim, who is a fan of TWiV and also a chronic fatigue, fibromyalgia, whatever patient. So he's been that way for 15 years, and he likes science, and he has 2 suggestions.
Importantly, he has an employment history as a exploration geologist, river geomorphology,
really?
And then had a career in electrical engineering, designing and manufacturing ASIC microchips. So
wow.
He's not naive to science.
Yeah,
right.
His career ended when he developed his symptoms. I'm sorry, I didn't mean to cut that out. He likes science. He likes stories. I also think one of the more important problems in our society today is that the common person is not educated in science, and the more science education we can give people, the better they can understand our complex society and its trials and tribulations. So we're his heroes as well as other podcasters. So that's great. 2 suggestions. This is for Alan. Number 1. Please listen into the recent TWiV podcast with Dr. Singh. I think, Alan, you were on that podcast.
I was.
Contrast her and Vince's responses to the listener's email versus your responses. Then toss me an email back or give me a phone call with your thoughts. I really don't think either of you are well educated on the history of CFS and the government research agencies. It's an unbelievable story. The following is by no means a complete history. It's just some tidbits that you can use to partially educate yourself or use to dig further into the story. So he provides a series of links which on the history of chronic fatigue syndrome /ME. So there's an ABC video. There is a history in the Times. And there's the involvement of the CDC in this. And his basic premise is that there have been starts and stops in research on this syndrome, and he feels that it hasn't gone as it should have been. For example, if you dig into it, you will find all sorts of twists and turns in the saga of federal government research monies and chronic fatigue, and the truly sad thing is it hasn't changed. I also suggest that you check out Dr. Judy Mikovits of the WPI, her statements concerning her desire to share samples. From what I can tell, most of the other researchers looking for XMRV, with the exception of Dr. Singh, ignore Dr. Mikovits. And I'm sure you remember from your TWiV interview, she said XMRV is very hard to find. And then he complains about the chronic fatigue name, which we talked about last time. It was another email asking us not to call it
right,
chronic
and I would
fatigue.
disagree. He says everyone associated with CFSME, the patients, the researchers, the clinicians, etc., all absolutely hate the term chronic fatigue syndrome. I would disagree with that. The reason that I use it is that it is in fact commonly used in the research literature. So the researchers'
preferred term.
It depends on who you ask, as far as I can tell. Some prefer this CFS/ME designation. Some prefer fibromyalgia. There are a number of papers that use that title. So ME standing for myalgic encephalitis. The issue here I would like people to understand is that I don't mean anything by choosing the CFS term. It's a convenient abbreviation to use, and that's the only reason. It appears a lot in the research literature, and this disease has many names, so that's the one that I happen to use.
Well, he says that if we keep using it, then everybody else will, but we use what
we're using it because everybody else is.
But as we said last time, if there's an etiology identified, the name will probably change.
That's right.
And we will change it too, but we're not there yet. And we're just here to talk about the science, not argue about the nomenclature. Oh, actually, we did argue about nomenclature, didn't we?
And in fact,
we'll argue about nomenclature, but only if it's about viral nomenclature.
But a ton of viruses have as their name where they were discovered. And so I wonder how this will play out once they find out where the first case was.
I just am not qualified to talk about the nomenclature of chronic fatigue syndrome /ME.
Yeah, I'm not either, which is why I picked one abbreviation and decided I would stick with it.
I don't mean any insult to anyone, but I don't know about it. And when it becomes a viral disease, if it does, then we will name it as such.
Right.
The bottom line on all these links, I actually looked at all these, and the bottom line on all these links is that there is what they all say. I won't judge try and make a judgment as to whether or not it's true. But what they all say is that basically the complaint is that the CDC in particular and other research agencies also for the longest time have not taken this disease seriously, and that even to the point of misappropriating some funds intended for research on this disease. And all I can say is that if that ever were true, I hope that time is over, and that people take it as seriously as they can and try and get to the bottom of it. If you look at the CDC site, what's written there suggests that they are taking it seriously. I hope that's true. And if it is, let's move forward.
Hear, hear.
Yeah, and I actually, in my early days as a science reporter in the late 90s, I covered the story on the CDC funds appropriation little scandal that they had. And there was definitely, they definitely sent some funds to the wrong account, and there was a dispute about why that was done. So that's not made up. That's an actual case where there was a misappropriation. Some of the folks said this was a paperwork error, and some folks outside the agency said that this was part of a pattern. Depends on where you sit on the issue. My own feeling is that government agencies should not put funds in the wrong places. These funds were assigned for CFS, and that's where it should go. And that was remedied, as a matter of fact. That when the problem was discovered, that was set back the way it should have been.
Well, as Rich says, let's move forward.
Exactly.
CFS-ME is getting much more attention now because there's a potential virus. We'll see where that ends up. But even wherever it ends up, the disease is now quite prominently, if not understood, discussed at a different level than it was before, and that can only be good.
And we should also reiterate again that in looking into the science, as we have done on this show, it's important to understand that it's not as if at this point it is established that XMRV is the cause. This is still an open subject. We're still trying to figure this out, and it's going to take a while.
Yeah. In fact, there have been a couple of papers lately, but we haven't talked about them. But I think maybe we'll do a roundup in a couple of weeks and just summarize what's going on. And just to wrap up Tim's email, thank you for taking the time to read this. As I said, I'm a fan. I'll still stay a fan.
Good.
I've learned so much from your podcast. The world needs more like it. Educating the public about science is so important. Sometimes educating the science about the public is necessary. Also amounts to what I was hoping to do with this letter/email. So all of it will be put on the website, Tim, and thanks for writing it. And let's do one more, which is from listener John, who made a drawing of Alan.
Yes.
I'm taking Vincent's call for a group drawing, so here is my Alan Dove. This was drawn in a 21-inch Wacom Cintiq, which is somewhat more accurate than the iPad. Okay, now a word about beards. They are a pain to draw, so please give Rich my regards as the least facial-haired PhD on the show. Seeing as I am going to redraw Vincent and sketch Dick, I'm looking forward to draw Rich, well, as he appears on the About page anyway, so I'll put up the photo of Alan. That's okay with you, Alan, right?
That's fine with me. I do not mind my caricature being...
Yeah, they are caricatures, aren't they?
They are.
Yeah.
That's right, they are.
And Alan said he looks evil and he loves it.
Yes.
He wants to be evil. Let's do a couple of picks of the week. Hey, Dickson, do you have a pick of the week?
I certainly do, Vince. I've got a summary pick of the week from the Centers for Disease Control of the final case count for West Nile virus for the year 2010. We're at the end of the season. We've been at the end of the season for quite a while now, so the final figures are in, and it turns out that we only had 964 total cases this year. And we had, in terms of numbers of deaths, 41. We had non-neuroinvasive disease cases of 375 and neuroinvasive disease cases 589. The epicenters for the virus infection this year were the American Southwest, particularly Arizona and California and New Mexico. We had some pockets of infection around Chicago. And we had, Rich, we had your area of the world invaded by West Nile virus quite heavily. Florida had a total of 11 cases. When I say heavily, that's certainly a drop in the bucket compared to what we've had in the past. And all of it is related to the presence or absence of drought. I went back through the USGS website and looked up the drought watch patterns for those same areas where the cases occurred, and each one of those areas had severe droughts this summer. So there appears to be still holding the correlation between droughts and the occurrence of West Nile virus in people. So that's my pick of the week.
Very good. That's at the CDC?
Yep.
Oh, there we go. There's a link. Rich, maybe we can ask the mosquito guys, the bug guys about this.
Ooh, let's do that. Yeah, absolutely.
We'll be at Fort Myers, Florida next week for a TWiV, and we're going to talk about dengue in Florida.
I'm going to be in Naples next week.
Florida?
Yeah.
That's not far, you know.
I know.
And we have some bug experts from the Keys flying in for the TWiV. And
what day are you going to be there?
Monday.
Monday. Too bad. I'll be there on Thursday. On the 9th.
Well, if you want to Skype in, you're welcome.
I might.
Because Alan is going to be Skyping in. I'll give you the info if you want to do that.
Okay.
Rich, what have you for us today?
I have another feed from my wife. It's a New York Times article called The Mystery Red Bees of Red Hook.
Okay.
And since we've talked about bees here before and that kind of stuff, I thought this was kind of cute, if nothing else. So there are a bunch of people various people in Brooklyn who are amateur beekeepers.
Right.
This is called, urban farming, Dickson?
Indeed it is. It's all part of the same deal.
And some of these people were dismayed to find that the bees were making red honey that had a kind of a metallic taste to it.
Uh-oh.
And they became suspicious that the bees were visiting a local maraschino cherry plant. And went to the extent, actually, of having somebody do some chemistry on the honey and finding out that, yes, indeed, it was loaded with red dye number 4.
Oh, my goodness. Oops.
And so they don't have they don't have the smoking gun yet. All right. But all of the evidence points to the notion that the bees are visiting the effluent from this maraschino cherry farm and sucking up high fructose corn syrup laced with red dye 40 and bringing it back to their hives and making this strange red honey.
So there is a maraschino factory in Brooklyn?
Yeah. Dell's Maraschino Cherries Company over on Dykeman Street in Red Hook.
Red Hook. Now you know why it's called Red Hook.
So
how funny.
And this red honey, what do they do with it? Do they keep it for themselves?
As a shade of Robitussin.
Do they sell?
No, well, yeah. I mean, I think these are amateur farmers, so I don't know that they sell the stuff. I don't think you would be able to sell this except as maybe a novelty.
Did they eat the red honey? Or were they worried about it?
Well, they've at least tasted it because they said that it has a sort of a metallic taste to it.
I see.
But I don't know if they're consuming it regularly.
Nice story.
And apparently the bees coming back have this red on them. I love this last quote in the article. One of these bee farmers who has this problem said, when the sun is a bit down, they glow red in the evenings. They were slightly fluorescent, and it was beautiful.
I don't know. The fluorescent part is a little.
The fluorescent
scary.
bees coming back to the hive in the evening.
So, do we still have beekeepers listening? If you're there, tell us what you think about this.
Yeah, good idea.
We had a couple. Alan, what do you have for us?
I think I've picked this particular webcomic before as a pick of the week, XKCD, but I wanted to specifically highlight the one that just came out today.
It's great.
Which is
that's quick. I would say that's very quick.
He has just a great turnaround time on this, and it's a very nice synopsis of the relationship between science and the media, I think.
Yeah, why don't you... Tell us.
Yeah, so it's about the arsenic-based life discovery, and the scientists are gathered around getting ready for their press conference and saying our arsenic-based DNA discovery is cool, but these reporters are expecting life on Titan. Our press conference will be such a letdown. And so they ultimately they're looking for ways to make the press conference more entertaining, and they see online there's a suggestion to make something more entertaining. You could serve cocktails and hors d'oeuvres that fit the theme of your event. And so the last panel, we've got one of these scientists standing up at the podium, and the reporters are gathered around in front, clutching their throats and falling to the floor.
Excellent.
Pretty good.
Love it.
If you've never looked at XKCD before, I should say that you should also in each comic you should put your mouse over it, and a little pop-up text box will give you an additional punchline.
Oh, look at that! Yeah.
How funny.
According to a new paper published in the journal Science,
it's all footnoted.
Reporters are unable to thrive in an arsenic-rich environment. Excellent. Very good. That's wonderful. Thank you. My pick is an iPad app from PLOS. It's the PLOS iPad app on which you can read papers from all the journals.
Wow.
You know, PLOS Pathogens, Biology, PLOS One, Computational Biology, Underappreciated Pathogens, or whatever it's called. There's a name for it. I'm sorry.
PLOS Best Supporting Pathogens.
It's called...
Neglected disease.
Thank you. Neglected tropical diseases.
Yeah.
PLOS is
That's run by Peter Hotez.
I'm
huge
sorry. So this is a free app which you can use, and it's really nice. You can read any of the articles you want as PDFs. You can save them. You can annotate them. Really nice. So check it out. It's free, and they also have an iPhone app.
I already got it.
And
I
so
saw
this is
last night, and I downloaded it.
There you go. Check it out. I like it very much.
Wow.
And they've actually been able to do this. Someone did it for them for free. A couple of freelance developers. Tom did it. And he could do it because it's an open source platform as well. So check it out. And that will do it for another TWiV. A few notes for everyone. over at our website, TWiV.tv. I've been adding comments from iTunes. Those are always great, so check them out. We have a fan page on Facebook, facebook.com/This Week in Virology, all one word. We have hundreds of fans already. Go there. It's just cool. We post pictures and comments. You can put questions there if you want. Tell us if you have something you want us to discuss. You can put it there. Just a nicer way to get to know us for those of you who are on Facebook. Of course, you can always listen to TWiV at iTunes at the Zune Marketplace. If you do, why don't you subscribe? Leave some comments on iTunes to keep us on the front page of the Medicine Podcast section, where we have been for a good amount of time now. So thanks to all of you who have done that. You can also listen to TWiV on your smartphone. There is an app from microbeworld.org which can do that. It'll stream the episodes to your iPhone or iPad. There is now an Android version for your Android device, and I'll post the link for that. So all of that is at microbeworld.org. And, of course, if you go to twiv.tv, you can play the episodes there, download them, and also check out all of our wonderful show notes. Right, Dickson?
Right, Vince.
As always, send us your questions and comments to Dickson at... No, sorry.
Please.
TWiV at twiv.tv. Alan Dove is at alandove.com. Good to have you again.
Always a pleasure.
And don't forget, we'll be Skyping you on Monday from Florida.
Yes.
Hopefully that will work.
Okay.
Rich Condit is down in Florida where the gators get wet. Thanks, Rich.
And they got whacked also, unfortunately.
Yeah, it's okay.
Is the season over?
Yeah.
Yeah.
Oh, boy, is it.
So now we don't have to talk about it until next year.
Good.
We can turn our attention to the round ball.
Yeah, always good to be here, and I'm really looking forward to our little road trip. That'll be fun.
So I'm flying down to Gainesville on Sunday. Rich and I'll be staying with Rich Sunday night.
Nice.
And we'll drive Monday morning to Fort Myers.
Great.
It's a 4 or 5 hour drive.
Yeah, it is a little bit
And
more.
then we do a podcast for a virology course there, which we will record. We're going to have the bug guys flying in from Key West, and they'll join us. And we'll talk about dengue in Florida.
And the releases in Grand Cayman Island, I hope.
Yes, we want to talk about that too. Yes.
Oh, yeah.
And I think, Dickson, you ought to join us.
I will.
You could really make a difference.
I'm going to be working from home that day.
Do you have a headset yet?
I bought it. It hasn't come. I bought it through Amazon. It should be at my house.
Because you're a little echoey without it.
I'm sorry to tell you that, but I am.
Well, it's
I'm
not you.
I'm a little empty-headed to begin with.
No. It's just the technology. If you talk at your monitor with no headset, it gets echoey. But even with the echo, we will take you.
Well, if you're going to Florida, maybe you'll loan me this one to take home with me. Then I can.
Well, that connector won't work with your.
Well, hopefully it'll come by then.
I hope it comes tomorrow.
Yeah, me too.
Or today.
Or maybe it's waiting for me today.
Perhaps. Anyway, good to see you as well. Dickson is at verticalfarm.com.
Yep.
And
things are looking up.
Things are looking up. The Vertical Farm.
No, I'm serious. We're on our way to Chicago next week. I hope we have some interesting news for you on December 8th with regards to that visit.
Excellent.
Looks very promising, gentlemen.
Very good. Hope to hear more.
Yep.
I am Vincent Racaniello, and I'm at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.